This is Unit 5C, Part 1. Now, in terms of contraction, which is what muscle does, the contraction of skeletal muscle fibers refers to the action of the myosin cross bridges, where the myosin head tilts up and binds to the the actin binding site on the actin myofilaments. Shortening occurs when tension is generated by the cross bridges exceeding the force opposing that shortening, and contraction ends when the cross bridges become completely inactive. Whatever tension was generated declines and relaxation of the muscle is induced. Okay, at the organ level, remember that skeletal muscle does not have cells but has muscle fibers, and each muscle is an organ in itself. The contraction of the individual muscle fibers and the muscle is similar. So, what I say in terms of the contraction applies to the entire muscle also, not just individual skeletal muscle fibers. There are two types of muscle contraction. One is isometric and the other is isotonic. Iso referring to same, and metric a measure of distance, and tonic a measure of tension. So an isometric contraction involves increasing in muscle tension, but the muscle length does not shorten at all. It does not change at all. There's no shortening during that contraction. And that's what you do when you tense your muscles but don't move any parts of your skeleton. For example, you tense your arms and your muscles will increase in their tension, but there's no shortening of the muscle. There's therefore no movement of any part of your body. Isotonic contraction is different, and you have to separate these two types of contraction. Isotonic contractions, you get a change in muscle length. The muscle shortens, but you don't get any change. You don't get any. You apply tension by shortening the muscle, and that usually remains more or less the same, or it ramps up, and that's mostly staying the same. The thing that's changing mostly is the muscle length, and this is what happens when you contract a muscle and you get movement of part of the body. So in one case you get a muscle being tensed but no shortening, and in the other case you get muscle shortening but no a force applied that's fairly steady. I don't know how helpful that was, but we'll give examples of that later. Okay, when we look at nerve-muscle relationship and look at the whole relationship as a function, and that and try and define the functional unit, we look at typically a motor unit. And a motor unit involves or comprises one motor neuron and all the muscle fibers, skeletal muscle fibers that it innervates or supplies. The number of skeletal muscle fibers that you get in a particular motor unit can vary. It can be 4 muscle fibers, or it can be several hundred muscle fibers. The muscles that control very fine movements have very small motor units. The motor neuron in that motor unit is going to be innervating just a few skeletal muscle fibers, so that you can make very fine movements. And these are the kind of things that are associated with the muscles in the fingers and the muscles in the eyes. So here you see a muscle on the upper left, and the muscle is a single organ. It's cut in cross section. Inside the muscle you have the bundles of skeletal muscle fibers, which are fascicles. So you have several fascicles, and within each fascicle you have several skeletal muscle fibers. Now the innervation, I would remind you that when you do a cross section through the spinal cord, that the inner butterfly-shaped section is the gray matter, where the neuron cell bodies are, and the axons are going to be going out of and into through the dorsal root here. Here's the dorsal root ganglion, a collection of the nerve cell bodies of sensory nerves or sensory neurons, which send signals into the spinal cord. Okay, so here we have signals going in from sensory neurons, sending signals in, and the signals will go into the dorsal root, which is here. Oh, sorry, the dorsal horn, and will make connections with motor neurons in the on the ventral side, in the ventral horn area, in here, in this area here. This is where the motor neuron cell bodies are, and they can send, if activated, they can send signals out. So here we see, let's follow one motor neuron cell and its axon going out through the ventral root and going out the the vertebral spines, or the spinal nerve, and going to some muscle fibers in a muscle. And here you can see that it's innervating two, at least they're showing two, it's probably innervating more muscle fibers. Here's another one, and that's called motor unit 1. And here's another one, I'll do it in another color. There's the cell body in the dorsal and the ventral horn, and there's the axon going out through the ventral root, and there's where it innervates three different skeletal muscle fibers within a muscle. These skeletal muscle fibers are not, that are innervated by any one particular motor neuron, are not all bunched in one area. In one area, they're spread throughout the muscle so that you get an even kind of shortening as motor units are activated and recruited to carry out a signal that leads to contraction. Remember that a motor unit can be anywhere from 4 to several hundred. It can innervate in a motor unit, you would have anywhere from 4 to several hundred skeletal muscle fibers as part of that unit. So, large weight-bearing muscles have large motor units, meaning that they have a motor neuron that is innervating quite a large number of skeletal muscle fibers. If it's finer motion parts of your body, your lips, your tongue, your eyes, it would be a smaller motor unit. The motor unit, as I said, the muscle fibers in the motor unit are spread throughout the muscle. They're not bunched in one area. The contraction of a single motor unit will cause, therefore, a weak contraction over the entire muscle, over the entire cross-sectional area of the muscle. Now, these diagrams here are, in fact, taken from experiments done using explanted muscles from experimental animals, where the muscle is suspended from a fixed one end of the muscle at the tendon is fixed, and the other one is suspended on or attached to a force transducer, so the amount of tension or force generated with the stimulus can be measured accurately. Now, the smallest possible contraction within a muscle is called a muscle twitch. So it's the response to a single brief stimulus that has reached threshold and triggers a contraction, the smallest possible contraction that can be induced. There are three phases characteristic of a muscle twitch. There's the first phase, which is a latent period. Here you see the stimulus, which is typically an electrical stimulus to the motor neuron innervating the muscle. The latent period is a period of very short period of few milliseconds following the stimulation, when during which excitation contraction coupling is taking place, and you cannot measure any tension generated because the process of excitation coupling has to occur. So here you see that there is a flat period where there is no tension generated. The next period is the period of contraction, in which the cross bridges between the thick filaments, the myosin thick filaments, and the actin thin filaments is forming, and the muscle is shortening by the repeated rapid ratchet-like attachment and detachment and shortening of the myosin and actin as they slide past each other. So here's the period of contraction, and then there's a period of relaxation during which calcium gets reabsorbed back into the sarcoplasmic reticulum, and the muscle tension goes back down to zero. So these are the three periods: latent period, period of contraction, and period of relaxation. Now, typically you get graded muscle responses. You can get a variation in the degree of muscle contraction depending upon how many motor units are going to be activated. And this is the kind of thing that is required for proper control of any movement of any part of your body. Will you move it more forcefully or less forcefully, faster or slower? Will you shorten the muscle and generate a lot of force by activating a lot of motor units, or just a few? The responses can be changed or graded. You can get a graded response by changing the frequency of stimulation or changing strength of the stimulus. So what you have is, for example, here in this kind of setup, the single stimulus resulting in the smallest single contractile response is called a muscle twitch. Okay, so a small stimulus, and you can generate these by increasing. You can do it experimentally by increasing the voltage used to stimulate a nerve, a motor neuron, or the muscle itself. It can be either stimulating on the nerve or stimulating onto the muscle. And as you increase it, eventually you hit the threshold required and you'll get a movement or contraction. You'll trigger excitation contraction coupling, and you'll trigger a small muscle twitch, a small shortening. Now, in this case, the example here is looking at increasing the frequency, not the size of the stimulus, but the frequency of the stimulus. And what's revealed by doing that is that you can get a thing called wave summation. So here you have a stimulus, and there's the latent period and period of contraction, and then the period of relaxation. And if you give another stimulus at some point before the muscle has finished relaxing, you will get another contraction, but this time a little bit stronger. And again, if you stimulate before it's relaxed completely, you'll get an even stronger contraction. This is called wave summation, in which the frequency is increased, but the muscle does not have enough time to relax. Now you can increase the frequency, and you can see this kind of effect ramping up, wave summation. This kind of, these are not the same, although they sort of overlap somewhat. But if you have continued contraction of a muscle, increasing frequency of stimulation causing a constant contraction, although the contraction gets stronger and stronger, that is called tetanus. Now this is an incomplete or unfused tetanus. If the frequency is so high that it's given before there's any relaxation at all, then that's called complete or fused tetanus. Tetanus is the term used for a particular disease that associated with a bacterial microorganism found in the soil called Clostridium tetani. Clostridium tetani causes release, so it will release a toxin that can affect the neuromuscular junction and cause continuous contraction of a muscle, and that's what tetanus really means, continuous contraction of the muscle. So the word is not strictly for disease, but also refers to this continuous contraction. Okay, so you can have wave summation, and it also shows the characteristic of muscles called tetanus when they are undergoing continuous contraction. Now, and that's explained, as I said, and written here, or more rapid frequency of delivery stimulus given very quickly without any allowing any period of relaxation. Now, as I said, when you stimulate, if you give too small a stimulus, you won't get anything. You won't trigger any kind of contraction at all. Once you hit threshold and you get contraction, that's the threshold stimulus required to see a first observable muscle contraction. If you stimulate at a higher level, then that's you'll get more vigorous contraction as the stimulus strength is increased. So this increase in contractions, more vigorous contraction as the stimulus is increased, is because more and more motor units will be activated. So if you're stimulating a nerve of a muscle, remember that the nerve carries the axons of the motor neurons, and by giving an electrical stimulus, you're short-circuiting things and inducing the signal to go down the nerve. You're acting using that electrical stimulus, you're mimicking a command that could come from a person's brain to send an action potential down a motor neuron. Now, as you increase the strength of the stimulus, you will activate more motor neurons and therefore activate more motor units. And it's the increasing strength of the stimulus which demonstrates that motor units can summate, and that's how we precisely control the force of contraction, by stimulating with our brain, a central nervous system, more or less intense commands, sending out a stronger command or weaker command to stimulate more motor units or less motor units, depending upon whether we need more strength or less. So this is called multiple motor unit summation, and is really motor unit recruitment. More and more muscle fibers are brought into play using this idea of recruitment. I would give you a very good example would be if you were trying to lift a 10-pound weight, and you had a good idea of the strength required, the weight of it, and how much effort you had to put in. Then you would send out enough commands to lift that 10-pound weight with your arms. You send out enough commands to the muscles in your biceps to contract. If then you're faced with a 1 pound weight, you would not recruit as many motor units. You would not send out as strong a signal through to the motor units, to the motor neurons, and therefore the motor units, and you would not recruit as many motor units. So recruitment is very important in precise control of contraction of a muscle. Okay, this is these are two graphs which are connected. One measures, they're looking at the same events. One measuring the stimulus strength, the stimulus voltage. Remember that this is a experimental setup where the muscle nerve is stimulated using voltage to mimic the commands coming from the brain. And at the same time, the tension is measured and shown in the bottom graph. Now, if you give a voltage stimulus which is too small to hit threshold, that's a subthreshold stimulus. And this is a subthreshold stimulus. We know it's a subthreshold because you don't get any tension generated in this, in these events, during this time period. If you give a stimulus that is just at threshold, you will get a recruitment activation of motor unit, which can respond and give a muscle twitch. So that results in a muscle twitch, the smallest stimulus that can trigger any contraction in the muscle. Once you raise it, you have to raise it enough to recruit, to result in recruitment of more. So this stimulus here is not changing the tension because it doesn't recruit another motor unit, but once you raise it high enough, you will get recruitment of a second motor unit, and therefore the force generated rises. And here, as you increase, you get an even larger generation of tension. It'll continue to go up until you get to a maximum tension generated because all the motor units are recruited. Down here, you shouldn't. This is a little bit. This here is a little bit misleading. It's really the size of the motor units. So it's a small number of fibers activated, or a medium number of fibers activated, or a large number of fibers activated when you're activating larger motor units, to larger, not so much in size, but in the number of skeletal muscle fibers that are within the motor unit. Okay, there is another characteristic feature of muscle contraction that you should be aware of, and that is called TREEP. I'm not sure if I'm pronouncing it correctly, but it's called TREEP, or the staircase effect. And that's an important concept to understand. The staircase effect can be demonstrated by giving repeated stimulus of the same strength immediately at the end of the period of relaxation. So as soon as the muscle gets relaxed, you give the stimulus. You don't change the strength of the stimulus, but you give the same stimulus, but you give it just when the muscle has gone through the period of relaxation. And what will happen is that you'll get an increased strength of contraction. The tension generally will be bigger because there still wasn't enough time for calcium to be, all the calcium that was within the sarcoplasm, the cytoplasm of the skeletal muscle fiber, to get pumped back into the sarcoplasmic reticulum, stored away before the next stimulus came. And so the stimulus that's given as soon as the period of relaxation ends will release more calcium into the skeletal muscle fibers, resulting in a stronger contraction because there's still some calcium left over from the last one. It wasn't all pumped back into the sarcoplasmic reticulum. The other thing is that typically enzymes, and muscle contraction involves generation of energy, and the generation of energy by oxidation of glucose and triggering of and activation of the Krebs cycle and generation of ATP by aerobic respiration in the mitochondria. It all involves quite a few different enzymes converting substrates to products, and then the product is the substrate of the next enzyme in the chain. And different enzymes have different activities at different temperatures. Fact is, as the energy gets generated, it's not 100% conversion of ATP to energy being put into the tissue to allow it to contract. Some of the energy is lost as heat, so heat is generated. So there is, as you know, when you exercise and you use your skeletal muscles, you generate heat. Now that heat, in part, it's not good if it's excessive, but in part is beneficial because many of the enzymes will increase in their activity with an increasing amount of heat. And so with repeated stimulation of muscle, there is more heat generated, and the efficiency of the system increases because enzymes will increase in their efficiency of converting substrates to products, and so more force is generated as the muscle contracts. And here in the next slide, you see this kind of effect where you have repeated stimulation given just as the period of contraction, period of relaxation ends, and it's very carefully timed. This is not the same as recruitment, which is demonstrated by giving a stimulus during the contraction, before the period of contraction ends. This is immediately as the period of contraction ends, you give the same stimulus, same size, these are all the same size stimuli, and you get a stronger contraction each time, up to a maximum. Of course, it won't get stronger and stronger unlimited, so it hits a plateau. Now this kind of staircase effect, in which there's increasing calcium availability and increasing temperature, making enzyme systems more efficient. This is the kind of thing why people talk about warming up before doing a exercise. And this is, there's a much more scientific meaning to the idea of warming up than most people understand or imagine. It literally does mean they're warming up. They're warming up their system, their muscles. They're getting the enzyme systems to be more efficient by that warming up. So warming up really is extremely important before an intense, prolonged muscle use. All right. Let's talk a little bit about muscle tone. The tone of a muscle is the constant background, slightly contracted state. And I don't know if you recall, but we already talked about autonomic tone, in which you have a certain amount of muscle tone in the smooth muscles, for example, around the blood vessels, so that, if they're partly contracted, then that's kind of muscle tone is important, because then they can either relax and you can get vasodilation, or they can contract and you can get vasoconstriction. You can go either way. If they were fully relaxed, then you wouldn't be able to get any more vasodilation. If they were fully contracted all the time, a lot of muscle tone, then you wouldn't get any more vasoconstriction. So a certain background muscle tone is important. It is also not only for autonomic nervous system responses, commands to muscle, cardiac muscle or smooth muscle, but also in the somatic nervous system, the commands to the skeletal muscle. So good muscle tone is useful and is really an indication of good health. It helps to keep the muscles firm, healthy, ready to respond. It doesn't produce any active movement, but you have a slight contraction of the muscle. So it's not fully relaxed or fully contracted at rest. the reason that you have these background muscle activity or muscle tone, slight shortening of the muscle, is because of spinal reflexes that are constantly activating one motor unit after another throughout the skeletal muscle structures. They're in fact responding to sensory signals coming from activation of stretch receptors in the muscles and the tendons. Remember the muscle spindle and the Golgi tendon organ in the tendons. Okay, let's. I said we would get back to the idea of isotonic versus isometric contractions. In isotonic contraction, the muscle changes in length. The angle of the joint across which the muscle is connected will decrease. So, for example, if you contract your biceps, it'll shorten and your lower arm will bend upwards. The angle decreases. And the load, if you have something in your hand or even just the weight of your lower arm, the load gets moved. There are two types of isotonic contractions that you should consider. One is concentric and the other is eccentric. In a concentric contraction, the muscle will shorten and does work, such as if you hold a weight. Let's pretend that, forgive me for making such a crude diagram, but let's pretend that this is a weight in somebody's hand. There's their lower arm and there's their upper arm. And if they did a concentric contraction, what you would get is there's their upper arm and their lower arm is now raised up, and there's the weight that they raised. So you got this movement along in this direction. Now that would be a concentric isotonic contraction. The muscle shortens and does work. An eccentric contraction is different. There is work being done. The muscles are being activated, but the muscle lengthens. And the example I would show you is when you have, instead of going in this direction, in this case, which is concentric, I'll put a C here for concentric. I'll put it in a different color for clarity. Concentric contraction. If, on the other hand, we go to a different example, what you have in an eccentric contraction is that the person's arm extends downwards. The muscle actually is lengthening. So here, and there's the weight. So what you got, in fact, was a movement in this direction, right? But it wasn't just relaxation of the muscle. It was giving force, but not enough force to lift the arm, but to slowly, in a controlled manner, lower the arm out. So you are doing work, you are using your muscle. The muscle does contract, but it will lengthen. The reason is you're not contracting with enough force to counter the weight. So you are doing work, but it's a different kind of work. It is an eccentric contraction. So those are the two types of isotonic contractions that are possible. This is a diagram that shows you some of the characteristics of an isotonic concentric contraction. Here you see a muscle set up so that it is connected at one end to weight down here through its tendon, and at the other end it's fixed, and there's presumably a force transducer that measures the amount of tension that's generated. So the tension that's developed, in this case, of course, if it rises up, it's going to be 3 kilos of tension, right? With stimulation of the muscle shortens. In other words, its resting length it'll decrease from its normal resting length. It'll get shorter, and that's what's happening starting there, going down here, and the weight is raised up. So you're generating tension and shortening the muscle. That's a concentric, isotonic concentric contraction. Now the other kind of contraction that we need to consider are isometric contractions, and I'll remind you, in this case, it's a question of there's no movement. The muscle is tensed. Force is being generated, but there's no movement and there's no change in the muscle's length. It neither shortens nor lengthens. And this will occur if the load is greater than the level of tension that the muscle is able to develop. For example, if you have a weight sitting on a desk and you try and lift it with your hand, but you can't generate enough force, so the load is greater, the load sits there, it doesn't move, your arm muscles are tensing and developing force, but not enough force to lift the load. And so you do contract, but there's no movement, so that's an isometric contraction. And this is shown in the following diagram here, where the 6 kilo weight does not move. There's no change in the length of the muscle. There's no overall change in length of the muscle. The weight doesn't move, although there is tension generated. Notice that the tension is less than the actual load. So the tension generated is not 6 kilos. If it was 6 kilos, then the weight would move upwards because the muscle would shorten. So here you have the kind of thing that happens when you tense your muscles, but there's no movement other than the muscle shortening, but none of your limbs or bones, there's no movement in any joint. And this is the kind of thing that people are told to do isometric exercise or contractions when they're sitting for a long time on an airplane, not moving. The reason for that, the reason they're told to do that is so that there is some squeezing of blood vessels, particularly veins, to help move the blood along. Otherwise, they sit for too long and the blood may become not be moving, returning back along the veins back to the heart fast enough. So there's an increased chance of developing a thrombus in those veins where the blood is essentially pooling. So people are told, particularly if you're older, if you have any problems with blood clotting, told to do, if either stand up and walk around the plane, or if you can't do that, then at least do isometric exercise, contract your muscles sitting in your chair, not moving. So let's move on to a different topic. Let's talk about the metabolic activity in muscles, which is responsible for generation of energy and different sources of that energy. In the end, the only energy available will be from the ultimate source is going to be ATP. So any system that generates ATP any of those systems are pathways or metabolic biochemical pathways that will provide energy for the muscle. The muscle needs ATP to contract, right? Now, the available sources of energy are as follows. Initially, there is a little bit of stores or reserves of ATP in muscle, but it's quite small and it is rapidly used up. It's rapidly hydrolyzed. So within the first 4 to 6 seconds of a muscle contracting, whatever ATP was there before the contraction started will be used up. Then it has to be regenerated. the most rapid way of regenerating ATP is through the interaction of ADP with a compound or molecule called creatine phosphate, abbreviated CP. Now in this reaction, what happens is that the phosphate from the creatine phosphate is donated to the ADP to form ATP. I remind you that ADP stands for adenine diphosphate and ATP stands for adenine triphosphate. The other way that ATP, there are two other major pathways that allow for ATP to be generated. One is through anaerobic glycolysis, the breakdown or oxidation, the breakdown of glucose in the absence of oxygen, or aerobic respiration, which involves oxygen for generation of ATP. We'll go through these in some detail. So here you see the three sources, pathways that lead to ATP production. Remember, there is also the ATP that's already in the muscle, the stores that are there, which are immediately used up. And then there's direct phosphorylation of ADP by creatine phosphate. Creatine phosphate donates the phosphate to ADP to form ATP. There's no use of oxygen. There's 1 ATP produced per creatine phosphate, and the duration of this source of energy is limited. It can be exhausted or used up within the first 15 seconds of contraction. So this is a short-term, what's called actually a substrate level phosphorylation, because it's a reaction that simply involves the donation of phosphate from creatine phosphate. So this is substrate level phosphorylation, where this is a substrate interacting with the ADP to form ATP. I would remind you that creatine then goes on to actually leave the cell. Some of it leaves the cell as creatinine and then gets excreted in the urine. Now let's go to C before we do. Let's go to C. Aerobic respiration or aerobic metabolism. In this case, we have, and I remind you, and you should have covered this previously in the last semester, that glucose, the carbohydrate glucose, is often used as a source of from which energy can get produced. It is broken down through a process of oxidation, and it goes through a number of different pathways before energy gets produced. But it's this kind of aerobic metabolism or aerobic respiration is quite efficient at producing a lot of ATP. For every single glucose molecule that's broken down, you get 36 molecules of ATP, which is a lot better than 1 ATP per creatine phosphate that was produced in this direct phosphorylation or substrate level phosphorylation reaction. Now the pathways involved, they're just mentioned. They point out here this that your livers and your muscle also actually store glycogen as a storage form of glucose. Remember, glycogen is made up of thousands of molecules of glucose all strung together. So you have glucose in your muscle in the form of glycogen, and it can draw on that storage form, or it can get it from the bloodstream. If glucose is low, glycogen is low in the muscle, then it can draw glucose from the blood. If glucose is low in the blood, glycogen from the liver will supply glucose to the blood, which then supplies it to the muscle. So glucose is there and gets broken down by the process of glycolysis. I hope you remember that. glycolysis. The first pathway here is glycolysis. First step in the pathway here is glycolysis. And actually, well, I should put it here also. This is what we're talking about, C here, glycolysis. And glycolysis leads to production of pyruvic acid. And pyruvic acid then gets converted to acetyl-CoA, and that goes into mitochondria, and you get the Krebs cycle. Krebs cycle produces ATP, but also products from these pathways, glycolysis, pyruvic acid, oxidation of pyruvic acid, and the Krebs cycle. They all also produce molecules that will be used in the mitochondria to generate, with the help of oxygen, to generate a lot of ATP. Okay, so this is oxidative phosphorylation, or aerobic respiration, or aerobic metabolism, generating ATP. The byproducts, it uses oxygen, and it needs oxygen. Remember, oxygen was the final electron acceptor in the electron transport chain. It produces some water that's called metabolic water, and all these pathways also produce CO2, which is why you breathe off CO2 as a waste byproduct of this system. Now, that kind of generation mechanism to generate ATP can provide a source of ATP for hours. If you're a trained athlete or do regular exercise, you can exercise for not too intensely, but you can exercise for hours and hours. You know, people who are cross-country skiing or long-distance running, things like that. Now, this mechanism, which starts with glycolysis, requires to go through aerobic respiration. You need, in the end, oxygen as a final electron acceptor. If you're running low on oxygen, then the muscle tissue will go to a different kind of mechanism to generate ATP, and that is anaerobic or lactic acid metabolism. So in this case, when there's not enough oxygen being supplied, then glucose will undergo glycolysis, and you get generation of pyruvic acid as an end product of glycolysis, but it doesn't go on to be used for it doesn't go on into the mitochondria to be used in the Krebs cycle or electron transport chain. Instead, it's used to produce lactic acid. So this is lactic acid fermentation, and as a byproduct of that lactic acid fermentation, 2 CO2 molecules are produced per glucose. That's a 1 per glucose molecule that's broken down in this way. So there's no oxygen being used, 2 ATPs per glucose, and the other thing is that the end product is not only ATP but also lactic acid. Now, this kind of energy source, it says 30 to 60 seconds, but the truth of the matter is that in any kind of endurance or a long-term aerobic respiration or activity, exercise, if you're exercising and you're going on for quite a while, and then you start exercising too intensively, so your cardiovascular and respiratory system can't provide enough oxygen to the muscles, you're using it up too fast, then you will start switching over. It's not a on and off kind of thing. It doesn't mean that you have a sudden turning off of aerobic respiration and then switching on of lactic acid fermentation. It's a gradual process. So, if you did have zero aerobic this kind of metabolism going on, then you could only sustain this for 30 to 60 seconds. But it's actually a sort of a transition over, so they overlap somewhat. But strictly anaerobic lactic acid fermentation on its own, you can't continue doing it for more than about 60 seconds. So knowing that, then you have a good feeling for the kind of, hopefully from what I've just told you, you have a good feeling for the kind of transition of energy sources that occur as you exercise. When you start at first, you have a few seconds, very short, 2 to 5 seconds, or whatever I said before, 2 to 6 seconds of ATP stores in your muscles that you're using. Then you use up the creatine phosphate to do direct phosphorylation, and then your aerobic respiration is working and you're producing lots of ATP. If you run out of glucose, then you would, well, I'm not going to get into that, but if you do it really intensely exercise and you exceed the ability of your systems to provide oxygen to the muscle, then you'll go into anaerobic respiration. And if you're doing only anaerobic, you only have, a very short, up to say a minute that you can do that. And eventually, then at that point, you'll stop from exhaustion, buildup of lactic acid, cramping, lack of energy. No energy, no muscle contraction. Okay. All right. And what I was telling you is some more details are here with anaerobic glycolysis, anaerobic respiration. That kicks in when the contractile activity of the muscle reaches about 70% of maximum. In fact, the contracting and bulging muscles will compress blood vessels and start to limit the amount of blood that can get into the muscle, so oxygen delivery is impaired, and you switch over from aerobic oxidation breakdown of pyruvic acid in the Krebs cycle and electron transport chain to lactic acid fermentation. So pyruvic acid goes to lactic acid fermentation and you stop. Within about a minute after that. The lactic acid, once you stop, lactic acid rapidly diffuses out of the muscle into the bloodstream, and it can go then to the liver, kidneys, and heart, but mainly the liver, where it gets converted back into pyruvic acid. Well, if you don't have ATP, then you'll go into a state of muscle fatigue, and it occurs when ATP production fails to meet the pace or the rate of use of ATP, or there's some sort of deficit of ATP that'll cause contractures, the continuous contraction of the muscle. Remember, ATP is needed to release the myosin heads of the thick filaments from the thin filaments. So if you don't have enough ATP, you can't get release, and the muscle is continuously contracted. There's also an accumulation with muscle fatigue. You get accumulation of lactic acid inside the muscle. It can't diffuse out into the bloodstream to go to the liver to get broken down. And you get ionic imbalances in the skeletal muscle fibers, because there's not enough time or energy available also for the pumping activity that would balance out ions properly. Those require some energy input, and so you get muscle fatigue and you stop contracting the muscle. You have to stop. Trained athletes who do not exceed the intensity that they're capable of for a long time, they don't exceed their aerobic abilities, can continue to exercise the muscle for long periods of time. I mean, they're these ultra marathoners who just keep going, but they're highly trained athletes who don't, but even someone who's not that well trained can still contract certainly a muscle for a prolonged period of time if their, motivation is high enough and they don't exceed their aerobic metabolic abilities. So very intense exercise will produce rapid muscle fatigue. Although once you stop, the recovery is pretty quick. When you have muscle fatigue, you don't have enough ATP, so it's not only a question of not being able to contract anymore, continue relaxing, contracting the muscle, but the sodium-potassium pumps cannot restore the ionic balances, as I mentioned earlier. Nevertheless, you can do low-intensity exercise for a long period of time, and that will you won't get fatigue for much longer. You have a much longer period that you can go before the muscle gets fatigued. During muscle fatigue, the sarcoplasmic reticulum can be damaged and oxygen regulation is disrupted within the skeletal muscle fiber. If you do this kind of very vigorous exercise, you get a dramatic change in the chemistry, and you have to recover from that. And the main thing is you go into oxygen debt. You don't have enough oxygen. The oxygen reserves have been depleted and they have to be replenished. So you stop and the oxygen reserves start to get replenished. The myoglobin in your muscle picks up oxygen. More oxygen is delivered into the muscle. It's able to build up again some base level oxygen and return to normal. Lactic acid diffuses out into the bloodstream and gets converted in the liver to pyruvic acid. Another important thing that has to be restored is that you've been using up glycogen stores in the muscles typically when you're doing very vigorous exercise, so you have to build that up again by eating and taking in enough carbohydrate to place that. And the baseline stores of ATP, which aren't huge but are in muscle, that has to be resynthesized, as well as creatinine has to be rephosphorylated. Phosphorus group has to be added to creatinine to make the creatinine phosphate, not creatinine, but creatine phosphate. So those reserves are resynthesized. This kind of deficit of oxygen it can be measured by how much oxygen is needed to restore you to the normal levels to carry out all these restorative processes. When muscle is active, you have heat production because only 40% of the energy generated or being used up is used for actual contraction and relaxation of the muscle to do actual work. There are about remember, no system is 100% energy efficient. About 60% actually goes towards the generation of heat. And if the heat level builds up then too high, then that's bad. You won't be able to function, but it's typically prevented by increased flow of blood to the skin so that you radiate away heat from your skin. And sweating is also a way of increasing the loss of heat. The heat will transfer to the perspiration on the surface of your body faster and also transfer to the air faster if you're sweating, perspiring. And that's the end of Unit 5C, Part 1.